Welcome back to the deep dive. I have to admit, today's topic has me looking at my coffee cup a little differently. We tend to think of our bodies as these solid, mechanical things, right?
We do.
Muscles pulling on bones like pulleys, the heart pumping like a mechanical engine. It feels very industrial.
It does. It feels structural. We look in the mirror and we see the hardware.
Exactly. But looking through the research we've pulled for today, specifically these massive physiology texts from Ganong and Gaitan, it turns out that's only half the story. If you really look under the hood, we are essentially walking, talking, bubbling chemical reactions.
That is such a profound shift in perspective. You're moving from anatomy, which is really just a map of the structures, to physiology.
And physiology is the logic.
It's the logic of how those structures talk to each other. It's the software code running the hardware.
And today we are looking at the body's wireless network. We're diving into the endocrine system. I want to start with that comparison because it was the first thing in the notes that made me go, oh, okay, I get it.
That's a great starting point.
We have two major control systems in the body, the nervous system and the endocrine system.
Right. And they're the yin and yang of control. They complement each other perfectly.
So if the nervous system, your brain, your spinal cord, is like a hardwired fiber optic cable delivering these instant pinpoint messages.
Like move this finger now.
Move this finger now. Or, ouch, that's hot. Then the endocrine system is. Oh, well, how would you describe it?
If the nerves are a landline or that fiber optic cable, the endocrine system is the Wi-Fi. Or to use a more modern analogy, it's a viral broadcast on social media.
A viral broadcast. Explain that. I like that.
Think about the mechanics. A nerve impulse is private. It goes from point A to point B. It's a direct text message to a specific muscle. Contract, boom, done.
One-to-one communication.
Exactly. But the endocrine system releases chemical messengers, hormones, into the bloodstream. That is the broadcast.
Oh.
Once it's in the blood, it goes everywhere. It travels to your big toe, your liver, your brain, your heart. It's a public post for the whole body to see.
But, and this is the part I was struggling with at first, if I release a hormone that's meant for my kidneys and it goes to my big toe, why doesn't my big toe freak out?
That is the beauty of the system. Even though the broadcast is public, only the followers pay attention.
Oh.
Only the cells that have the specific receiver, the receptor, for that hormone will actually read the message. The rest of the body just ignores it. It's just background noise.
So the pancreas might tweet out insulin and the whole body sees it, but only the cells with the insulin app installed actually do anything about it.
That is a perfect 21st century analogy for it. Precisely.
Right.
And this difference in delivery dictates the speed.
Right.
Nerves are milliseconds. Hormones. Well, hormones take their time. The blood takes about a minute to circulate fully.
So the endocrine system isn't for dodging a bus?
Not at all. It's for the long game.
The long game. What do you mean by that?
Growth, metabolism, reproduction, maintaining the salt and water balance in your blood. These aren't split-second decisions. They are processes that require persistence.
You don't want your growth spurts to happen in milliseconds.
That would be terrifying.
I'm just imagining it.
It would be messy. You need a system that can sustain a signal for minutes, hours, or even days. That is the domain of the endocrine system. And the ultimate goal of all this broadcasting is this concept of homeostasis.
Homeostasis. I remember that word from high school biology. It's the Goldilocks state, right? Not too hot, not too cold.
It is the golden rule of physiology. It's the maintenance of a stable internal environment. I mean, think about it. Outside your body, the temperature changes. You eat a bag of salty chips. You forget to drink water. You get stressed.
Your external world is chaos.
Complete chaos.
Yeah.
But inside, your blood sugar, your temperature, your hydration, they stay remarkably flat. The endocrine system is the thermostat, the regulator of the software running in the background, ensuring that despite the chaos outside, the inside stays steady.
So today, we aren't just going to memorize a list of glands. I feel like we all did that in school and promptly forgot which one is the adrenal and which one is the thyroid.
Oh, absolutely.
We are going to look at the logic of the system. We're going to figure out how a chemical signal made in the brain finds a specific cell in the kidney, why some signals act instantly while others take days, and what happens when this delicate system goes haywire.
And honestly, understanding this explains so much about your daily experience. It explains why you get thirsty, why you feel sluggish when you haven't eaten, how stress physically changes your metabolism, and the mechanics of massive diseases like diabetes. It's the operating system of your life.
Okay, let's unpack the cast of characters first. We keep saying hormones, but that's a broad term. It's like saying vehicles. A bicycle and a jumbo jet are both vehicles, but they work very differently.
That is a fair comparison. In the source material, they break hormones down into 3 main families based on their chemistry. And I know hearing chemistry makes some people want to tune out.
I was about to say, don't give me flashbacks to organic chem lab.
I promise to keep it painless. But the chemistry matters, because chemistry is destiny in physiology. If you know what a hormone is made of, you effectively know how it's built, how it travels in the blood, and how it talks to a cell.
Okay, I'm with you. Who is the first character in our lineup?
The first and largest group are the proteins and polypeptides.
Polypeptides, that just means chains of amino acids, right?
Yeah, exactly. Think of them as beads on a string. These range from tiny little peptides, maybe only 3 amino acids long, like TRH,
TRH being?
thyrotropin-releasing hormone, all the way up to large, complex proteins with nearly 200 amino acids, like growth hormone.
So this is the vast majority of hormones?
Yes. The hormones from your pituitary gland, your pancreas like insulin and glucagon, these are all proteins or peptides. They're water-soluble, which is a key detail we'll come back to.
Okay. Then we have the second group, which I think people are more familiar with, at least the name, the steroids.
These steroids. Now, chemically, these are completely different. They aren't chains of beads, they are lipids. Fats, essentially. And here is the kicker. They are all synthesized from cholesterol.
Which is a good reminder that cholesterol isn't just a villain clogging your arteries.
Not at all. It's an essential precursor. If you had 0 cholesterol, you would die. You literally need it to make testosterone, estrogen, cortisol, aldosterone.
The stress and sex hormones.
The backbone of your stress response and your reproduction, exactly.
And the third group?
The amine hormones. These are derivatives of a single amino acid called tyrosine. It's a small but powerful group.
And it includes?
It includes the thyroid hormones and the catecholamines epinephrine and norepinephrine from the adrenal medulla.
Epinephrine is adrenaline, right?
Yes, in the U.S. we usually say epinephrine, but it's the same thing as adrenaline, the fight-or-flight hormone.
Okay, so we have proteins, the beads, steroids, the fats, and amines, the tyrosine edits. I want to talk about how these are made because reading through the Ganong text, I got this image of 2 very different factory models.
That's a great way to think about it.
The peptide hormones seem like they are built on a classic industrial assembly line.
That's a very accurate analogy. Because peptides are proteins, they follow the standard protein synthesis pathway in the cell.
Let's break that down for the non-biologists. How does that factory work?
Okay, imagine the nucleus of the cell is the architect's office. It holds the blueprints, the DNA.
The master plan.
Right, and you want to make some insulin. You don't take the master blueprint out of the office, you make a copy. That copy is the messenger RNA, or mRNA.
So the courier takes the copy out of the office.
Right, and runs it to the factory floor, which we call the rough endoplasmic reticulum. It's rough because it's studded with little machines called ribosomes.
And the ribosomes read the blueprint.
They read the blueprint and start snapping amino acids together. But the text mentions something interesting here. They don't just make the finished hormone right away.
No, they make something called a pre-pro hormone, which sounds like the most bureaucratic name ever. I am the pre-pro manager.
It does sound administrative, doesn't it?
Yeah.
But it's necessary. The pre-pro hormone is a large, inactive chain. It's like a rough casting of a metal part.
So it's not functional yet?
Not at all. It includes the signal sequence, basically shipping label, that tells the cell, hey, sell me to the packaging center.
So it travels through the factory.
As it enters the endoplasmic reticulum, that shipping label gets clipped off, and now it's a prohormone, still inactive.
One step closer.
It is then shipped to the Golgi apparatus, the shipping and handling department. There, it gets packaged into secretory vesicles. These are like little delivery trucks.
And inside the trucks.
Inside those trucks, enzymes finally chop it up into the active hormone. Sometimes there are other fragments made too, which can be useful for diagnostics.
And does it move immediately?
No. And this is the crucial part for peptides. The cell keeps a stockpile of these vesicles, filled with ready-to-go ammunition, sitting right near the cell membrane. They're just waiting.
Waiting for what?
A signal. Usually a spike in calcium or something called CAMP. When the signal comes, say your blood sugar spikes, the cell doesn't have to start building insulin from scratch.
It's already made.
It's already made and packaged.
Yeah.
The cell just opens the loading dock doors. The vesicles fuse with the cell wall in a process called exocytosis, and boom, the hormone is released into the blood instantly.
So it's a store and release model. Massive inventory, ready to deploy at a moment's notice.
Exactly. It's high inventory, fast response. Now, contrast that with the steroid hormones.
But steroids don't seem to believe in inventory.
Not at all. Steroid-producing cells operate on a just-in-time manufacturing model. If you cut open a cell in the adrenal cortex, you won't find little bags of cortisol waiting to be released.
So what do they store?
They store the raw material. They have large vacuoles filled with cholesterol esters.
Okay, so they have the lumberyard, but they haven't built any furniture yet.
Correct. And this brings us to a really fascinating mechanism mentioned in the Ganong source. When the signal comes to make a steroid, say, your brain screams stress. The cell has to start building right then and there.
And there's a bottleneck.
There is. The bottleneck, the rate-limiting step, is getting that cholesterol into the mitochondria, which is where the first chemical conversion happens.
And that requires a specific bouncer, right? The STAR protein.
Yes, the steroidogenic acute regulatory protein, STAR. I think of STAR as the VIP security guard. The cholesterol is waiting outside the club, which is the mitochondria.
And it can't get in.
And nothing happens until the signal arrives and STAR shows up to escort it in.
So a signal hits the cell, STAR gets activated, drags cholesterol into the mitochondria, it gets converted to pregnenolone, and then processed into the final hormone.
But here is the problem with storage and why they have to be just in time. Remember, steroids are lipids. They are fat-soluble.
And cell membranes are made of fat.
Exactly. The cell wall is a lipid bilayer. If you made a steroid hormone and tried to put it in a vesicle, a bag made of lipid, it would just diffuse right through the wall of the bag.
And then right out of the cell.
And then right through the wall of the cell itself. It's like trying to hold water in a net. You can't contain it.
You can't. That's fascinating. I never thought about that physical limitation.
So nature's solution is, don't make it until you need it. As soon as a steroid is synthesized, it simply drifts out of the cell and into the bloodstream.
That is a huge difference. Peptides are build, store, dump. Steroids are signal, build, leak.
Which means peptide responses can be incredibly fast dump the cargo. Steroid responses take a little longer to ramp up because you have to start the manufacturing process from scratch each time.
Before we move on to how they travel, there was one more detail about release that I found surprising. We tend to think of hormones as a steady drip, drip, drip.
Like a leaky faucet?
Yeah, but the sources emphasize that it's often pulsatile.
This is a critical nuance, and one that is often missed. Hormones are rarely released at a constant rate. They come in bursts or pulses. It's oscillatory.
Like a heartbeat?
Similar concept, but much slower. Sometimes pulses happen every hour, sometimes every few hours. This is driven by clocks in the hypothalamus. The suprachiasmatic nucleus is the master clock, coordinating these rhythms.
But why does that matter? Why not just have a steady stream? It seems simpler.
It turns out the target tissues, the cells receiving the message, get bored easily.
They get bored. A cell gets bored.
Well, desensitized.
Yeah.
If you yell at someone constantly, eventually they just tune you out. That's downregulation, a concept we'll come back to.
Okay.
A steady, high level of hormone can cause the cell to decrease its receptors and start to ignore the signal. But if the hormone comes in pulses, signal, silence, the tissue stays fresh and responsive.
That has to be a nightmare for medicine, though.
It is.
Okay.
It's a huge challenge. Think about hormone replacement therapy or giving insulin. We usually give these things as a pill or a single injection.
Right, a smooth curve.
We are giving a flat, monotonous note to a body that is used to a complex, rhythmic song.
So we are blasting them with a steady signal, and the body might actually stop listening because of it.
In some cases, yes. It's one of the reasons why matching the body's natural physiology is so difficult in a clinical setting. We can copy the molecule, but it's really hard to copy the rhythm.
Okay, let's get these hormones on the road. Section 2 of our outline is the commute. Our hormone has been made, it's been released, pulsed out into the bloodstream. Now it has to get to work.
And we're back to the oil and water problem.
Always back to chemistry. The bloodstream is plasma. Plasma is mostly water.
It is.
So for the peptide hormones and the catecholamines...
It's an easy commute. They are water-soluble. They dissolve right into the plasma and float freely to their destination. They are like swimmers in a lazy river. They just go with the flow.
No special transport needed.
Nope. But the steroids and the thyroid hormones, they are the fatty ones.
They're lipids.
They are hydrophobic. They hate water. If you just dump testosterone into the blood, it would clump up like oil droplets in a vinaigrette. Or it would stick to the walls of the blood vessels. It wouldn't get anywhere.
So they need a ride?
They need a boat. We call them plasma carrier proteins.
And there are different kinds of boats, right? It's not just one generic ferry.
That's a great way to put it. There are general ferries, like albumin. Albumin is a protein made by the liver, and it's like a city bus. It will carry almost anything. It's not very specific.
Okay.
And then there are specific luxury yachts designed for one type of passenger. For example, sex hormone-binding globulin, or SHBG, specifically carries testosterone and estrogen. Transcortin specifically carries cortisol.
Now, this part blew my mind a little bit. When a hormone is sitting in the boat, when it's bound to the carrier protein, it's inactive. It can't do anything.
That's correct. To enter a cell and do its job, the hormone has to step off the boat. It has to be free. Only the free fraction is biologically active.
So why bind 99% of it? The text says thyroid hormone is more than 99% bound. Why have all this hormone floating around if it can't be used? It seems so inefficient.
Think of it as a savings account versus the cash in your wallet.
Okay.
The free hormone is the cash in your wallet. You spend it, you use it immediately. But if you only had the cash in your wallet, you'd be broke in an hour.
Right, you run out.
The bound hormone, the stuff on the boats, is the savings account. It's a reservoir. As the tissues use up the free hormone, or as the kidneys filter it out, the chemical equilibrium shifts, and more hormone unbinds from the carrier to replace what was lost.
So it maintains a steady supply of cash in your wallet.
Exactly. It buffers against sudden drops, and it also protects the hormone. A tiny free peptide floating in the blood is vulnerable. Enzymes chew it up, or the kidneys filter it out very quickly.
How quickly are we talking?
Let's look at angiotensin II from the Guyton text. It's a peptide involved in blood pressure. It floats free. Its half-life is less than a minute.
Less than a minute.
It's there, it acts, and it is destroyed. Gone.
Wow. Gone in 60 seconds.
Less. But thyroid hormone, because it is so tightly bound to carrier proteins, its half-life is 1 to 6 days. It lingers. This allows for that long-duration background regulation, keeping your metabolism steady week over week.
That makes total sense. So the carrier protein dictates how long the hormone survives.
It dictates its clearance. We call it the metabolic clearance rate. It's the balance between how fast you make it and how fast you remove it.
And removal is happening how?
Several ways. Metabolic destruction by tissues, binding to tissues, excretion in bile by the liver, or excretion in urine by the kidneys. The boats protect the hormones from being cleared too fast.
Okay, so the hormone has survived the commute. It arrives at the target tissue. Now we get to the really cool part. Section 3, the reception. This is how the cell listens.
And this is where the concept of specificity is paramount. Remember our viral broadcast analogy?
Yeah, the message goes everywhere.
The hormone is everywhere. But only the cells with the specific receptor for that hormone will respond. It's a lock and key mechanism. No receptor, no response.
But the cell isn't just passive, is it? It's not just a door with a lock. The cell can change its locks.
Exactly. The cell is smart. It manages its doors. If there's too much hormone flooding the system, like a loud noise, the cell can protect itself by downregulation.
So it just removes the receptors.
It physically pulls the receptors off the surface and destroys them. It stops listening. It's becoming desensitized.
But that's the nose blindness thing again. You walk into a room, it smells. 5 minutes later, you don't notice it anymore.
Exactly. Conversely, if there's very little hormone, the cell might upregulate, build more receptors to catch every distinct whisper of a signal. It becomes more sensitive.
Now, depending on what kind of hormone we are talking about, the receptor is in a different place. This is the location matters part of our outline.
Right. It goes back to solubility again. If you are a peptide or a catecholamine water soluble, you cannot cross the cell membrane.
Wall of fat.
The cell membrane is a wall of fat. You bounce right off, so you have to ring the doorbell.
Your receptor on the surface.
Your receptor's on the surface or membrane. You bind to the outside, and the message is passed inside.
But if you are a steroid...
You are a ghost. You are lipid soluble. You drift right through the membrane wall like it's not even there.
So your receptor's waiting for you inside.
Usually in the cytoplasm.
And the thyroid hormone...
Even deeper. It goes straight to the nucleus. Its receptor is practically attached to the DNA chromosomes themselves.
I want to slow down here and really visualize these mechanisms because the text threw a lot of acronyms at us. Let's start with the surface receptors, the doorbell method.
Okay.
The hormone rings the bell. It never enters the house. How does the message get inside?
Through a second messenger. The hormone is the first messenger. It hands the note to a butler inside the house.
And the most common butlers are?
The most common mechanism for this involves G proteins.
G proteins, I always visualize this as a Rube Goldberg machine. It's a chain reaction.
It absolutely is. Okay, picture this. The hormone binds to the receptor on the outside. The receptor changes shape. It physically twists.
Okay.
This twist kicks a G protein that is attached to the inside of the membrane.
The G protein is the next domino.
Right. The G protein drops a molecule of GDP and grabs a molecule of GTP. That's the switch turning on. When that happens, a piece of the G protein, the alpha subunit, breaks off.
And it goes somewhere.
It runs along the inside of the membrane to find an enzyme. One of the most famous is adenyl cyclase.
Adenyl cyclase, what does that do?
It's a factory for the second messenger. It takes ATP, which is the cell's energy currency,
Right.
and it converts it into cyclic AMP or CAMP.
And CAMP is the butler running through the house shouting the message.
Yes, CAMP rushes through the cell, activating other enzymes called protein kinases. And here's the most important concept: amplification.
Explain that.
One single hormone molecule binds to the outside. It activates one G protein, but that G protein can stimulate the production of hundreds of CAMP molecules.
Oh, wow.
And each CAMP activates a kinase. Each kinase then phosphorylates and activates hundreds of other enzymes.
So it's a cascade, an avalanche.
A massive avalanche.
Oh.
One molecule at the door triggers millions of actions inside. This is why you don't need gallons of adrenaline to make your heart race. A few picograms of dusting triggers a massive body-wide response in seconds.
That explains the speed. The surface receptor system is built for speed and power.
Precisely. And it's not the only one. There's another pathway mentioned, phospholipase C.
Right, the notes mention something about calcium.
Yes. This is another G protein path. Instead of making CAMP, the G protein activates an enzyme called phospholipase C.
Okay.
This enzyme splits a membrane fat, a phospholipid, into two second messengers, IP3 and DAG.
Sounds like a rap duo.
They act like one. DAG stays in the membrane and activates protein kinase C. But IP3 is the interesting one here. It floats into the cytoplasm and goes to the endoplasmic reticulum. Remember the factory?
Yeah.
And it tells it to release its stored calcium.
Why is that important? Why is calcium locked up?
Because usually, a cell keeps calcium levels inside very, very low. Calcium is a powerful trigger. It's almost treated like a poison that you keep locked away. If you flood a cell with calcium, things happen.
Like what?
Muscles contract. Vesicles release their cargo. Genes can be activated. It's a major go signal.
So when I get a stomach cramp or my blood vessels constrict, that's IP3 unlocking the calcium cage.
In many cases, yes. It's using the cell's own dangerous stockpile of calcium to trigger an event.
Okay, so that's the surface. Ring the bell, trigger an avalanche. Now let's look at the slow burn, the intracellular and nuclear receptors used by steroids and thyroid hormones.
This is a completely different logic. Since the steroid drifts through the membrane and finds its receptor inside the cytoplasm.
Right, it's already in the house.
It's in the house. The hormone receptor pair, now bound together, actually walks right into the nucleus.
Into the command center? To the blueprints?
It goes to the DNA. It binds to a specific section called a hormone response element. And it basically tells the architect, we need to build a new wing on the house.
It changes the blueprints.
It changes the transcription.
Yeah.
It directly initiates or alters the process of making new mRNA, which then goes out to the ribosomes to build new proteins.
That sounds slow.
It is slow. It takes time to unzip DNA, copy it, process the copy, send it to the factory, build the protein, fold the protein.
Yeah.
You can't fake that physics.
Right.
This is why if you inject adrenaline, your heart rate jumps in 3 seconds. But if you take a steroid for inflammation or testosterone for muscle growth, you don't feel it instantly.
It takes hours or days.
Because you are literally remodeling the cell. You aren't just turning on the lights, which is what the surface receptors do. You are building new furniture.
Remodeling the house versus turning on the lights. That is the perfect distinction.
And that's why Section 1, the chemistry, was so important. If you know it's a steroid, you know it's going to be slow, sustained, and involve DNA. If you know it's a peptide, it's likely fast, amplified, and involves surface switches.
All right, we've covered the who, the how, the commute, and the listening. Now we need to talk about the management. Section 4, the logic of control. Because if you have all these powerful chemicals floating around, you need a way to shut them off.
You absolutely need feedback loops. The system would run wild without them.
The text mentions negative feedback as the stabilizer.
Negative feedback is the bread and butter of physiology. It's the Goldilocks principle. Not too hot, not too cold, just right.
Can you walk us through the blood osmolality example from the text? I found that one really clear.
Sure. Let's say you're hiking, you're sweating, you haven't drunk water in hours, you are dehydrated.
Okay, been there.
This means the concentration of salt and other solutes in your blood, osmolality goes up, your blood is getting salty and thick.
Gross, but okay.
Specialized cells in your hypothalamus called osmoreceptors literally shrink because water is being osmotically sucked out of them. They fire a signal.
They sense the change.
You physically sense it. This triggers the release of vasopressin, also called ADH or antidiuretic hormone, from the pituitary gland.
vasopressin enters the blood.
It travels to the kidney. It binds to receptors there and tells the kidney, stop dumping water in the urine, save it. So the kidney reabsorbs water back into the blood.
Which dilutes the blood, bringing the saltiness back down.
And here's the feedback. When the osmolality drops back to normal, the osmoreceptors rehydrate and stop firing.
So they stop screaming for vasopressin.
And vasopressin release stops. The system shuts itself off. The response removes the original stimulus.
The response removes the stimulus.
Exactly. It loops back to negative the original signal. That's negative feedback. It's all about stability.
But then you have positive feedback, the surge. This sounds dangerous, like a runaway train.
It can be. Positive feedback is explosive. Instead of dampening the signal, the effect amplifies the signal. It pushes the system further and further away from normal.
Why would you ever want that?
You want it when you need a definitive, irreversible event, like childbirth or ovulation, things that need to reach a climax.
The outline mentions the LH surge before ovulation.
Right. In the menstrual cycle, as the follicle in the ovary grows, it secretes estrogen. Usually, estrogen has a negative feedback effect on the pituitary.
So it keeps things stable.
Yes. But at a certain critical level, just before ovulation, the switch flips. The high level of estrogen suddenly has a positive feedback effect. It stimulates a massive release of luteinizing hormone, or LH.
And the LH causes more estrogen.
And the estrogen causes more LH. It spirals up and up in a massive surge until POP. The follicle ruptures, the egg is released.
Into ovulation.
Once that event happens, the cycle is broken, estrogen levels fall, and the system resets to negative feedback.
So positive feedback is for climax events.
Exactly. But if it happens when it's not supposed to, like in certain types of shock or heat stroke, it can be a death spiral. It can kill you.
And layered on top of all this feedback are the rhythms we touched on earlier. The circadian variations.
Right. Your feedback loops aren't static. The set points change depending on the time of day.
What do you mean the set points change?
Well, cortisol is high in the morning to wake you up and mobilize fuel for the day. Growth hormone peaks during deep sleep to repair tissues. The normal level isn't a single number.
It's a wave.
It's a wave. It's like a programmable thermostat. It knows to lower the heat at night and crank it up in the morning. So normal depends on when. If I measure your cortisol at midnight and it's high, that's a problem. If it's high at 8 a.m., that's healthy.
This brings us to Section 5, when the system fails, endocrine disorders. The text organizes these into 3 categories: deficiency, resistance, and excess. This felt like a really useful way to troubleshoot the human body.
It's a diagnostic framework. If a patient is sick, you ask, is the signal missing? Is the signal being ignored? Or is the signal screaming too loud?
Let's start with deficiency, the missing signal.
This is often destruction of the gland itself. Type 1 diabetes is the classic example. It's an autoimmune tragedy. The immune system identifies the beta cells in the pancreas, the factories that make insulin, as enemies.
And it burns the factory down.
It destroys them. So you have the raw materials, you have the blood flow, but the factory is gone, zero insulin. You have to inject it exogenously to survive.
Or it could be a dietary lack, right, like goiter.
Right. That's a different kind of deficiency. If you don't eat iodine, your thyroid gland cannot build thyroid hormone. It has the machinery, it has the signal from the brain, but it has no bricks.
And the gland gets bigger.
The gland actually grows larger, a goiter, because the brain keeps screaming, make more hormone. The gland hypertrophy is trying desperately to trap more iodine, but it fails.
Then there is resistance, the ignored signal. This one feels very modern, especially with the rates of type 2 diabetes.
This is the mechanism that is most misunderstood by the public, I think. In early type 2 diabetes, the pancreas is working fine. In fact, it's often working overtime.
So there's plenty of insulin.
There is plenty of insulin in the blood. The signal is being broadcast loud and clear, but the cells aren't listening.
Why not?
The receptors are downregulated, or the intracellular signaling, that kinase cascade we talked about, is gunked up, often due to chronic inflammation associated with obesity. The message arrives at the door, but the house is a mess and can't respond properly.
So what does the pancreas do?
It shouts louder. It secretes more and more insulin to try to force the sugar into the cells. This is a state called hyperinsulinemia.
And eventually?
Eventually, the beta cells get exhausted, they burn out and die. Then you have deficiency on top of resistance. It's a physiological tragedy of trying too hard for too long.
Finally, excess, the screaming signal.
This is usually tumors. If a tumor grows in the pituitary and starts pumping out growth hormone, it doesn't care about negative feedback. It has no off switch. It's autonomous.
Resulting in gigantism.
If it happens in a child whose growth plates haven't fused, yes, they grow to 7-8 feet tall. Think Andre the Giant. If it happens in an adult, it's called acromegaly, where the hands, feet, and jaw grow larger.
But there is another weird cause of excess: autoimmune mimicry. Graves' disease.
This sounded like biological identity theft.
It is. It's fascinating and terrifying. In Graves' disease, the body produces an antibody that happens, by sheer bad luck, to be shaped exactly like TSH.
And TSH is?
TSH is thyroid-stimulating hormone. It's the signal from the brain telling the thyroid to work.
Okay.
But in this case, the antibody latches onto the thyroid's TSH receptor and turns it on in permanently.
So the thyroid thinks the brain is yelling, work, 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 24/7.
Exactly. So it pumps out massive amounts of thyroid hormone, leading to hyperthyroidism. The brain sees this excess and stops making its own TSH, so the real TSH level is 0. But it doesn't matter.
Because the hacker is in the system.
The hacker antibody is still there, jamming the pedal to the floor. The feedback loop is working perfectly at the brain level, but the hacker is bypassing it completely.
It highlights how the system relies entirely on shape and fit. If you can fake the key, you can hijack the house.
We're coming into the home stretch here. Section 6. Measurement. How do we actually know any of this? Because, as the outline notes, these hormones are in the blood in picograms.
A picogram is a trillionth of a gram. It's an almost incomprehensibly small amount.
The analogy I saw was finding a pinch of sugar in an Olympic-sized swimming pool. How did we ever figure out how to measure that?
For a long time, we couldn't. We could only see the effects. Someone was 8 feet tall or someone had a goiter. But in 1959, Rosalind Yallo and Solomon Burson developed the radioimmunoassay, or RIA.
Yellow won the Nobel for this, right?
She did, and deservedly so. The method is genius in its simplicity. It uses competitive binding.
Explain it to me.
Okay, imagine a game of musical chairs. The chairs are antibodies that are designed to catch a specific hormone, say, insulin. You have a limited number of chairs.
Okay, we have the chairs.
Now, in the lab, you have a bottle of radioactive insulin. You've tagged it so you can see it.
Oh.
You know exactly how much is there.
Yeah.
You mix that radioactive insulin with the sample of the patient's blood, which contains their natural invisible insulin, and you throw them both at the limited number of chairs.
So they have to compete for the seats.
Exactly, they compete. If the patient has a lot of natural insulin, that natural insulin will shove the radioactive stuff out of the way and take most of the chairs.
So when you check the chairs at the end,
If the chairs aren't very radioactive, it means the patient's natural insulin won the competition. That means they have a high level of insulin.
and if the chairs are glowing,
If the chairs are glowing with radioactivity, it means there was very little natural insulin to put up a fight, so the radioactive version got all the seats. Low natural insulin.
that is brilliant. You measure the invisible by how much it displaces the visible.
It revolutionized medicine. Suddenly we could see the sugar in the swimming pool.
Yeah.
Modern versions like ELISA use color changes instead of radiation, but the principle is the same.
But just measuring the level isn't enough, right? The stress test concept from the outline.
Right, because the system is dynamic. A single snapshot can be misleading. If I see a low hormone level, I don't know if the gland is broken or if it's just sleeping because it hasn't been asked to work. I need to poke it.
So you do a stimulation test.
Exactly. If I suspect you have adrenal insufficiency or Addison's disease, I don't just measure your cortisol. I inject you with ACTH, the brain's signal to the adrenal.
You ring the doorbell and see if anyone answers.
Precisely. If your cortisol spikes up after the injection, your adrenal gland works fine. The problem is probably upstream in the brain. If it stays flat, the gland itself is dead.
And if you suspect excess,
You do a suppression test. The opposite. I give you something that should shut the system down via negative feedback. For cortisol, I can give you a powerful synthetic steroid called dexamethasone.
and that should tell the brain we have enough. Stop.
It should. A normal person's cortisol will plummet. But if the levels stay high,
It means the gland, or more likely a tumor, is ignoring the stop signal. It's gone rogue.
you essentially try to break the feedback loop to see if it's working.
Yeah.
In physiology, you learn more by stressing the system than by watching it at rest.
This has been massive. We've gone from the molecular assembly line inside the cell to the delivery trucks, the boats in the blood, the doorbells on the surface, and the remodelers in the nucleus.
It is a lot, but I think if you hold on to that broadcast analogy, it all holds together. The body dumps a signal into the river and relies on the intelligence of the receivers to keep us alive and stable.
And the balance of speed versus duration. The nervous system handles the immediate crises. The endocrine system handles the long game. the background processes.
And it does so through these beautiful self-correcting feedback loops. It's a system constantly trying to return to center, to that homeostatic balance.
It really makes you think about your own body differently. You aren't just a static object. You are and I'm quoting our notes here, a complex conversation of chemical probabilities.
That's a poetic way to put it. But it's true. A shift of a few picograms of testosterone or thyroid hormone can change your mood, your energy, your body shape, your entire personality. We like to think we are in control of our minds, but often we are at the mercy of these molecules.
So here's my final thought for the listener to mull over.
Right.
We talked about circadian rhythms and how the environment inputs signals into this machine.
Right.
In our modern world, bright blue lights from screens at night, constant low-grade stress from notifications, endless food availability, we are sending our endocrine system signals it wasn't really designed to handle.
We are hacking our own system.
Yeah.
And not always in a good way.
Mm.
We are inputting daytime signals at midnight. We're providing feast signals constantly and famine signals never.
Exactly. So maybe think about that the next time you're scrolling your phone at 2 a.m. or grabbing a sugary snack. You're not just making a choice. You're inputting data into a very sensitive, very ancient biological algorithm.
Yeah, and the algorithm will respond. It has to.
Thanks for listening to The Deep Dive. We'll catch you on the next one.